Hey students, this is your Nandini Ma'am. Today we are going to continue our reading from 3.4 Biochemical Processes in Cells, Module 1 Cells as the Basis of Life from Biology in Focus Year 11 revised. So let's get started. Eukaryotic cells contain many membrane-bound organelles that contain specific enzymes that catalyze specific biochemical processes. For example, chemical reactions in lysosomes break down compounds brought into the cell by using strong digestive enzymes in an acidic environment. Having the lysosome enclosed in a membrane prevents the enzymes from destroying the cell. These reactions within cells require energy, which is provided in the form of ATP. The two major biochemical processes that cells carry out are photosynthesis in plant cells and cellular respiration in all living cells. Energy transfer between reactions. Provision of energy is vital to ensure that all essential life processes take place. All cells use glucose as the primary source of energy to drive the thousands of chemical reactions that occur constantly in each living cell. When glucose is broken down in the process of aerobic cellular respiration, the large amount of energy contained within it is released. This released energy is trapped and stored in small packets in many high-energy molecules called adenosine triphosphate ATP. The cell assesses the energy contained in ATP to power all cell functioning. ATP is a small mobile molecule that can deliver the energy to wherever it is required. ATP is composed of a complex molecule called adenosine, adenine attached to a ribose sugar group, that in turn attaches to three phosphate groups. This is where the name adenosine triphosphate comes from. The bond that attaches the third phosphate group is a high energy bond. When energy is required by the cell, this bond is broken and the energy it contains is released. ADP and a free phosphate group are formed. To store the energy produced by the cell, a free phosphate group is attached to the ADP molecule by a high energy bond, forming ATP. The storage and release of energy is a cycle between ADP, free phosphate groups, and ATP, as shown in figure 3.34. Photosynthesis is the process by which plants utilize light energy, usually from the sun, which is trapped by chlorophyll contained in the chloroplast. It uses this energy to break apart water and carbon dioxide molecules and build them up into oxygen, energy storing glucose molecules, and water molecules. Photosynthesis can be summarized by the word equation carbon dioxide plus water in the presence of light energy and chlorophyll gives us glucose plus oxygen. The balanced overall chemical reaction is 6CO2 plus 6H2O in the presence of light energy and chlorophyll gives us C6H12O6 plus 6O2. The process of photosynthesis is summarized by the journal equation above. The actual process of photosynthesis occurs in two stages, each consisting of a series of many chemical reactions that take place in the chloroplasts of green plant cells and the cells of some photosynthetic bacteria. Phase 1. The light-dependent stage. The light-dependent stage, photolysis, occurs in the grana of chloroplasts. It involves the absorption of light energy by the chlorophyll in the thylakoid membranes of the chloroplast. This energy is used to split the water molecules into hydrogen ions and oxygen. The oxygen is released into the atmosphere and the hydrogen ions are carried to the next phase. ATP is also formed at this stage. Phase 2. The light-independent stage. The light-independent stage, which is carbon fixation, occurs in the stroma of the chloroplast. It involves combining in the light-independent stage the carbon dioxide form, which was there with the hydrogen ions produced in the light-dependent stage to form glucose. The ATP formed in the light-dependent stage is required to provide the energy for this reaction. No light is required in this stage. Glucose produced by photosynthesis can be converted by plants into complex carbohydrates, lipids, and or proteins. These end products are stored by the plants and are the source of organic nutrients not only for the plants themselves, but also for the consumer organisms that eat the plants. Key concepts. Energy in cells is transported within cells by small and mobile ATP molecules. ATP stores energy in a high energy bond that attaches the third phosphate group to the ADP molecule. When energy is required, the high energy bond is broken, releasing energy of phosphate group and ADP. Photosynthesis is the process where plants use light energy trapped by chlorophyll to break down water and carbon dioxide molecules and build them up into oxygen, glucose, and water molecules. Photosynthesis is a series of smaller reactions occurring across two stages. The light dependent stage occurs in the grana, where light energy splits the water molecules into oxygen and hydrogen. The light independent stage occurs in the stroma, where carbon dioxide and hydrogen combine to form glucose. Cellular respiration. All organisms break down glucose as a source of energy to drive cellular metabolism. Glucose can be broken down either in the presence of oxygen, aerobic cellular respiration, or in the absence of oxygen, anaerobic cellular respiration. Different products and amounts of energy are released in each of these processes. Anaerobic cellular respiration. Many organisms, such as some bacteria and archaea, live in environments lacking oxygen. Cells such as muscle cells sometimes cannot get enough oxygen, but still are able to release energy via an anaerobic pathway in the cytosol of cells. Two commonly used biochemical pathways enable energy to be produced using molecules other than oxygen: alcohol fermentation and lactic acid fermentation. In alcoholic fermentation, yeast, many organisms, and plants carry out anaerobic cellular respiration. This process involves the breakdown of glucose in the absence of oxygen to form ethanol and carbon dioxide. Two molecules of ATP are produced as a result. Glucose gives ethanol plus carbon dioxide plus adenosine triphosphate. Humans make use of this process in the production of beer, wine, and bread. Lactic acid fermentation is carried out when an organism cannot produce enough energy by aerobic cellular respiration. This occurs in periods of strenuous exercise when our body cannot deliver enough oxygen to the muscles to produce the energy required. In lactic acid fermentation, one molecule of glucose is broken down in the absence of oxygen to produce two molecules of lactic acid as well as two molecules of ATP. Once the strenuous exercise has finished, there will be more oxygen available. The lactic acid changes to a compound called pyruvate and is converted by aerobic respiration into carbon dioxide, water, and energy in the form of ATP. Aerobic cellular respiration. Aerobic cellular respiration is a chain of many biochemical reactions that occur in the cells of the organism in the presence of oxygen. It is generally summarized as follows to depict the main changes that occur during respiration. The word journal equation for aerobic cellular respiration would be expressing as glucose plus oxygen. Many reactions will be taking place and they'll give carbon dioxide plus water plus energy. There are at least 20 separate reactions, each catalyzed by a specific enzyme that makes up this overall pathway. The first step in the biochemical pathway to break down glucose is the process of glycolysis. This process occurs in the cytosol of the cell and involves the breakdown of glucose into two pyruvate molecules. This is accompanied by the release of energy in the form of two molecules of ATP. This overall reaction is actually a series of 10 smaller reactions, each catalyzed by a specific enzyme. The two molecules of pyruvate formed from one molecule of glucose then enter the mitochondria of the cell, where the rest of the series of reactions that make up this aerobic cellular respiration occur. Each of these reactions is catalyzed by a specific enzyme and accumulates in the production of more energy, carbon dioxide, and water. The energy from these reactions is released in the form of 34 ATP molecules to make a total of 36 ATP molecules produced for the breakdown of every glucose molecule. This process is summarized in figure 3.37. Photosynthesis and aerobic cellular respiration. Photosynthesis and aerobic cellular respiration are related. When you look at the journal equations for photosynthesis and aerobic cellular respiration, it appears that these processes may be the reverse of each other. That is not the case because they are each made up of a series of chemical reactions that are very different. Photosynthesis and aerobic cellular respiration are, however, closely related and interdependent. The products of photosynthesis are used in the process of aerobic cellular respiration. Similarly, the products of aerobic cellular respiration are used in the process of photosynthesis. In cells that contain both chloroplasts and mitochondria, the two processes will occur in the same cell. In cells that do not contain chloroplasts, such as plant root cells, the products of photosynthesis must be supplied to the cells for the process of aerobic cellular respiration to occur. Removal of cellular products and waste. The products of the reactions in the cell that are not required by the organism are called waste. They need to be transported to other cells and or need to be removed for efficient cell functioning. Removal from the cell will usually occur by diffusion through the cell membrane. Oxygen and carbon dioxide gases are easily removed from the cell by diffusion through the cell membrane. Any excess water that is not required by the cell moves out of the cell by osmosis. Waste formed from the breakdown of proteins also leave the cell by diffusion through the cell membrane. Waste chemical products that dissolve in water are removed from the cell at the same time as the water. Oily, fat-soluble substances are attached to small electrically charged molecules by the cell to make them water-soluble so that they can be removed when the water diffuses out. When parts of the cell break down, wear out, or die, they also need to be removed. Lysosomes, which contain digestive enzymes, are responsible for breaking down these old cell parts and other cellular waste. Any of these waste that can't be eliminated in this way by the lysosome are packaged and removed by exocytosis. Proteins such as hormones and enzymes and other substances such as mucus are produced by an animal cell. These cellular products are packaged into vesicles and leave the cell by the process of exocytosis. Wastes that leave a cell are said to be excreted from the cell. Useful products that leave a cell to be used elsewhere are said to be secreted. Key concepts. When glucose and oxygen combine in the process of aerobic cellular respiration, energy is produced along with water and carbon dioxide. This process is made up of many reactions. The energy stored is in the form of ATP molecules. The first step of this process occurs in the cytoplasm and is called glycolysis. In this step, one glucose molecule is broken down into two pyruvate molecules and two ATP. The second step in the reaction occurs in the mitochondria, where pyruvate in the presence of oxygen is broken down to release carbon dioxide, water, and 34 ATP. A total of 36 ATP molecules is produced for every molecule of glucose broken down in aerobic respiration. Anaerobic cellular respiration produces ethanol and carbon dioxide in bacteria, yeast, and plant cells, and lactic acid in animal cells. Two ATP molecules are produced in this process for the respiration of each molecule of glucose. Wastes are the products of cellular reactions that are not required. Many of these wastes can be removed by simple diffusion through the cell membrane. Other wastes can be removed by exocytosis or destroyed by the enzymes present in lysosomes. So that was the chapter. See you with the next chapter soon. Thank you.